We often think of bacteria as these microscopic self-contained units, right? But imagine within those tiny cells, there's another even tinier world of components that could be playing surprisingly huge roles, especially in how these bacteria live and interact with larger organisms like us and plants. We're talking about small open reading frames, or smORFs, and the mini proteins they code for called SEPs. It's almost like discovering a secret language within a language.
That's a really good way to put it. For so long, the spotlight in genetic research has been on the genes that make bigger, more obvious proteins. There was this kind of implicit cutoff. If it wasn't a certain size, well, it probably wasn't a real functional protein.
Yeah, like assuming only large buildings matter in a city and ignoring all the vital smaller shops and homes.
Exactly.
And because these smORFs are so small, I mean, we're talking sequences coding for proteins maybe just a handful to, 70 amino acids long. They've been incredibly challenging to find and to study. Our usual gene hunting tools often just skipped right over them.
Pretty much. And the resulting tiny proteins themselves are tough to detect in the lab. Very difficult.
Okay.
But, with advances in sequencing and proteomics, our ability to read DNA and analyze proteins, we're now realizing that this previously overlooked world of small molecules might be absolutely critical. Critical for understanding how bacteria function and, importantly, how they interact with their environment.
So in this deep dive, we're going to explore a fascinating recent study that has really dug into this hidden universe of smORFs. And what makes this research really compelling, I think, is its focus on bacteria that have these close relationships with eukaryotic hosts, that's us, and plants. So our mission today is to pull out the key insights from this study. They looked at a massive collection of annotated bacterial genomes to try and understand what these tiny genetic bits might be doing in the microbes sharing our world.
Yeah, and what's really impressive about the study is just the sheer breadth of it. They didn't just focus on one or two, model organisms. They cast a very wide net, analyzing annotated smORFs in a huge variety of bacteria, from ones crucial for plant health like rhizobia to pathogens that cause disease in animals. It gives us a much more panoramic view.
Okay, so before we get into the study specifics, let's just make sure we're all on the same page. What exactly are smORFs and SEP in this context? And maybe more importantly, why should you, our listener, really care about these seemingly tiny bits of genetic code?
All right, think of it like this. A genome is the instruction manual for a cell. Right. Within that manual are chapters, the genes providing blueprints for proteins, the cell's workhorses. Historically, we focus on the long chapters. But smORFs, they're like very short paragraphs, maybe even just a sentence within those chapters. They're open reading frames that, despite their size, can be translated into these small but potentially mighty SEPs, smORF-encoded polypeptides.
Right.
And why care? Well, because these tiny molecules are increasingly linked to fundamental bacterial processes, especially those influencing how they interact with other organisms.
So these aren't just genetic junk or noise then. These little SEPs are actually doing things.
Oh, absolutely. We're finding they're involved in a surprising range of activities, even though they've been under the radar.
Yeah.
We're talking about helping bacteria withstand stress, harsh conditions, contributing to antibiotic resistance, how they get nutrients, even fundamental stuff like forming spores or cell division.
Wow.
But the catch is, most of what we know in detail comes from just a few well-studied lab bacteria. This study really aimed to broaden that view, looking at bacteria living closely with plants and animals. And the fascinating idea here is that these tiny components could be key to understanding the very nature of those interactions, good or bad.
Okay, so how did these researchers even start to tackle this? Sifting through all those genomes for these tiny smORFs, trying to figure out which ones matter. It sounds like looking for needles in a haystack, a really, really big haystack.
Well, their approach was quite systematic and heavily computational, as you'd expect. They started by downloading a huge set of complete, high-quality reference genomes from the NCBI RefSeq database.
That's like the main library for this stuff, right?
Exactly, the gold standard. So they had a solid foundation. Then they used specialized computer programs to find all the annotated smORF sequences, specifically those predicted to code for polypeptides of 70 amino acids or fewer. And they gathered any existing info about predicted functions, though, as we'll see. That was often pretty limited.
Right. So they get this massive list of short potential protein coding bits. But how do you tell the important ones from just random sequences? How did they figure out which of these tiny genes might actually be doing something useful?
That's the crucial step. and their key innovation was assessing evolutionary conservation. The basic idea is if a particular smORF has been kept in the genomes of related bacteria over evolutionary time, it's much more likely to have an important function.
Makes sense. If it was just noise, it'd disappear or change quickly.
Precisely. And to get a really robust measure of this conservation, they didn't just rely on one method. They used a consensus approach, employing 3 different pangenome analysis programs: OrthoFinder, Panaroo, and OrthoGNC.
A consensus from 3 different programs. Why the extra work? Seems complicated.
Think of it like getting a second and third opinion. Each program uses slightly different algorithms, different ways of comparing genes across genomes. By seeing where they all agree, or mostly agree, on which small genes are present in related bacteria, well, you can have much greater confidence that those genes are truly conserved.
Okay. So it boosts the reliability.
Right.
Less chance of a false positive.
Exactly. It gives a more reliable picture of which smRFs are consistently present and therefore likely important.
So how do they categorize this conservation then, based on these 3 programs?
They created 6 categories. These ranged from probably not conserved all the way up to conserved, meaning it was found in all the genomes they looked at within a specific group. But one category became particularly important for their analysis: conserved 50.
Conserved 50.
Yeah, this included SAPs found in at least 50% of the genomes within a given bacterial group. It's a threshold to identify smORFs that are widely kept, but maybe not absolutely essential in every single strain. Their final decision on a smORF's conservation level was based on how well these 3 programs agreed. Strong agreement meant they adopted that level. More disagreement meant they were more cautious.
Okay, that sounds like a really thorough, thoughtful approach. Now, the study spent quite a bit of time on Synorhizobium mellolati as a kind of model system.
Right.
Why that one? And what do they find when they look closely at its smORFs?
Right. Synorhizobium mellolati. It's a well-studied bacterium, forms a symbiosis with plants like alfalfa, helps fix nitrogen, super important ecologically. Because we already know a fair bit about its biology, including some small proteins that have been experimentally studied, it was a perfect test case for their methods.
A proving ground.
Exactly. They found a typical S. mellolati genome has around 285 annotated smORFs, give or take. And interestingly, the majority of the predicted SAPs were between 40 and 70 amino acids long. They found very few annotated ones shorter than, say, 10 or 20 amino acids.
Huh. So does that mean there just aren't many ultra small ones? Or
Are current methods just not very good at finding and annotating them? That's the million-dollar question, really. It highlights a potential blind spot.
Yeah, that's a great point. The tools we use definitely shape what we see. What else stood out in S. mellolati?
Another key finding, a big majority, about 2/3 of these annotated SAPs were labeled hypothetical proteins.
The classic. We know it's there, but we have no idea what it does.
Exactly that. And what's even more striking, that proportion of hypothetical proteins was even higher for the shorter SAPs. It really just underscores the vast unknown territory we're dealing with here.
So a lot of mystery boxes. Were there any SAPs in S. mellolati where they had a better handle on the function?
Yes, thankfully a few. The study mentioned a small percentage with known roles. Things like cold shock proteins.
Which help bacteria survive the cold, presumably.
Right. And they also found some SAPs with a specific domain called DUF1127, which seems to be involved in nutrient acquisition. These known examples, though few, are crucial anchors for understanding what the others might be up to.
And how did their conservation analysis, that conserve 50 idea, compare to the SAPs in S. mellolati that scientists already knew existed from lab experiments? That seems like a critical check.
It absolutely was, a critical validation step. They compared their conserve 50 category, those found in at least half the S. mellolati genomes, with a set of 91 SAPs already experimentally confirmed. And encouragingly, their conservation analysis showed high sensitivity. It correctly flagged a large majority, over 90% of those known validated SAPs as being conserved.
Wow, okay, so that really backs up their approach.
It does. It gives us much more confidence that evolutionary conservation is a reliable filter. If a smORF has stuck around in related bacteria, chances are it's doing something important.
That's strong. Now, given that so many are these hypothetical proteins, what did the study do to try and, lift the veil a bit, get some clues about function?
Right. They didn't just stop at hypothetical. They used a range of bioinformatic tools, computational detectives, if you like to predict functions for the conserved hypothetical SAPs in S. mellolati. They used programs like Panzer2, which infers function based on sequence similarity to known proteins. They also looked for known protein domains, predicted if they might be stuck in membranes, transmembrane regions, or if they had signal peptides, which often means they get secreted or sent somewhere specific.
And did these computational detectives crack any cases? Did they manage to assign potential identities?
They got some interesting leads, yeah. For instance, one hypothetical SEP looked like it might be an integrase, an enzyme involved in DNA insertion. Another was predicted to have a Pepsi-Y domain, often found in proteins that inhibit other enzymes, proteases. They also found several potential transmembrane proteins and ones with signal peptides, hinting at roles in transport or communication.
But still just predictions at this stage.
Yeah. Exactly. The researchers were careful to point out that while these are valuable starting points, we really need methods and databases specifically tuned for these very short peptides.
Right.
Our current tools were mostly built with bigger proteins in mind, so functionally, we're still in the early days with these tiny molecules.
Okay, so S. melliloti was the test bed.
Yeah.
But they also looked at other bacteria where SAPs have been experimentally found, right? Did the patterns hold up elsewhere?
Yes, they did. They extended the analysis to several others with existing experimental data like Yersinia pestis, the plague bacterium,
Oh, wow.
Stutzeromonas stutzeri, Campylobacter jejuni, a common food poisoning bug, and Bradyrhizobium diazoefficiens, another nitrogen fixer. And across these different species, they generally saw the same trend. SIBC that were known from experiments tended to pop up in their conserved 50 category.
Okay, so consistency across different types of bacteria. That's good.
Yeah, it strengthens the idea that conservation is a pretty reliable indicator of a SIP being real and functional, broadly speaking.
Were there any sort of surprising findings or interesting quirks in these other bacteria?
A couple of things stood out. In Yersinia pestis, they found that many of the experimentally confirmed SFPs actually started at different points in the genetic code using alternative start codons compared to how they were annotated.
Meaning the gene prediction was slightly off.
Potentially, yes. And often these alternative starts resulted in slightly longer proteins. It suggests our standard gene finders might be missing some of these, especially the unconventional ones.
Interesting.
They also found that some validated SFPs that didn't meet their conserved 50 criteria did have similar sequences listed in a specialized database called SOR-FDB. This hints that even if a SEB isn't widely conserved across a whole group, it might still be expressed and important in specific species or conditions.
Right, so not conserved doesn't automatically mean not important or not real.
Exactly. It adds a layer of nuance.
Okay, so after these focus looks, the study zoomed out again, right?
Okay.
Looking across a whole range of bacteria associated with plants and animals. What were the big picture trends?
Generally, across many different bacterial genera, the number of annotated SFPs per genome typically landed in that 100 to 300 range. There were exceptions, sure. Bradyrhizobium and Bacillus often had more. Chlamydia had significantly fewer. They also confirmed their earlier finding. Most annotated SFPs in these diverse groups were longer than 40 amino acids, and that high prevalence of hypothetical proteins, around 60 to 70%, held true across most genera they looked at. The mystery is widespread.
A vast sea of we don't know yet. What about the conservation levels across these broader groups? Was that consistent and did it vary a lot?
It varied quite a bit, actually. Some groups, like Mycobacterium, showed a high proportion of conserved SFPs. Over 60% were in that conserved 50 category.
Suggesting they might be core functions for mycobacteria.
Potentially, yeah, fundamental roles. But then other genera, like Pseudomonas, had much lower conservation levels for their annotated SFPs, less than 50%. This likely reflects their different lifestyles and ecological flexibility. Bacteria with more specialized host interactions might rely on a more conserved set of small proteins for those roles.
That makes intuitive sense. Now, the study also tried to see if there were functional differences between SFPs in plant-associated versus animal-associated bacteria. What do they find there?
Yeah, that was a really cool part. They used a tool called EGNO-GMapper to assign functional categories, COG categories, to the conserved 50 SFPs in both groups. Again, a large chunk had unknown functions in both. No surprise there. But looking at the knowns, the top predicted functional categories were actually quite similar in both plant and animal groups.
Oh, like what?
Things involved in translation-making proteins, transcription reading the code, intracellular trafficking, moving stuff around, ion transport, energy production. basic cellular machinery. It suggests small regulatory proteins might be commonly involved in tuning these fundamental processes, regardless of the host.
But were there any hints of specialization, functions more common in one group than the other?
Yes, they did find some intriguing distinctions. For example, SAPs related to FLPFAP pylon components think bacterial hairs for attachment seemed more frequent in plant-associated bacteria.
Suggesting a role in sticking to plants.
Plausibly, yeah. Roles in colonization or interaction. In the animal-associated bacteria, they saw more of a protein domain called DUF645. Its function is still unknown, another hypothetical. But they did find certain SAPs and domains that were either exclusive or much more abundant in one group. It strongly hints that these tiny proteins might be tailored for interacting with specific host types.
Fascinating. So different toolkits of tiny proteins for different hosts.
Yeah, it looks that way. While some things like ribosomal proteins were shared, their patterns differed, suggesting fine-tuning even for common roles.
Okay, so as we start to wrap up our exploration of this study, what are the main takeaways you see about this hidden world of bacterial smORFs?
Well, this study really provides a valuable, broad survey. It reinforces that most bacteria likely harbor hundreds of these smORFs, typically encoding peptides in that 40 to 70 amino acid sweet spot. It also spotlights that lingering question about the scarcity of annotated SAPs under 40 residues. Is it biology or our methods? We still don't know for sure. A key conclusion is that evolutionary conservation, like their conserved 50 metric, is a really powerful indicator that a smORF is likely real and functional. They also noted that species-level analysis seems more sensitive for finding them, while genus level might be more specific for identifying broadly important ones.
And that point about the alternative start codons you mentioned earlier, that seems really important for how we find these things in the future.
Absolutely. It highlights a need to refine our annotation methods to catch these non-standard cases. And of course, the study just hammers home the huge gap in functional characterization. Most are still hypothetical.
Still mostly black boxes.
Their work showed we can start predicting functions bioinformatically, even if we need better tools designed for short peptides. And finally, finding SAPs and domains seemingly specific to plant or animal interactions, that opens up really exciting avenues for future research into host microbiology.
So bringing it all together, this deep dive shows us bacterial genomes are just teeming with these tiny smORFs, these polypeptides. They're likely playing incredibly important roles in how bacteria interact with hosts, plants, animals, maybe us, but also feels like we've only just started to understand what they're actually doing.
That sums it up perfectly. The challenges are significant, no doubt, but this research gives us a big step forward, a framework, and highlights so many potential candidates for future investigation. It's charted a lot of the unknown territory.
It really makes you think, doesn't it? Could these small, often ignored proteins be the key to understanding, maybe even manipulating complex biological interactions, like the balance in our own gut microbes or how crops interact with soil bacteria? What could we unlock if we could fully decode their functions?
Precisely. The potential implications for medicine, for agriculture, for just basic biology are potentially enormous. It really underscores that idea. Size isn't everything in biology. These small players could have a massive impact.
Absolutely. A real aha moment, realizing these tiny parts might hold some of the biggest secrets. And on that thought-provoking note, we'll wrap up this deep dive. Thanks for joining us as we explored the intriguing and often hidden world of smORFs.